Good morning, students. Today we are going to discuss culture media and culture methods. तो culture media के लिए basic constituents जो requirement होती है वो है basically water, electrolytes, peptone, meat या yeast, extract, blood, serum भी use कर सकते हैं and especially the agar which is used for the solidification purpose. Mineral salts जो कि add होते हैं they can be sulfates, phosphates, carbonates, sodium, potassium, chloride, magnesium and calcium. pH कल्चर मीडिया को बनाने के लिए इज अप्रोक्सीमेटली 7.2 to 7.6 एंड एसेसरी ग्रोथ फैक्टर्स कई बार रिक्वायरमेंट होती है फॉर द ग्रोथ ऑफ स्पेसिफिक बैक्टीरिया. फॉर एग्जांपल ट्रिप्टोफैन इज एडेड इन द कल्चर मीडिया फॉर द ग्रोथ ऑफ साल्मोनेला टाइफी एंड 10 and 5 फैक्टर फॉर हीमोफिलस इन्फ्लुएंजा. So now we are discussing the properties of agar. जो agar होता है वो extract करते हैं from the seaweeds. It consists of long chain polysaccharides, inorganic salts and small amount of protein like material. ये basically melt करता है at 98 degree centigrade और हम इसको solidify कर सकते हैं at 42 degree. Not degraded by microorganisms and ये does कोई भी nutrient provide नहीं करता. It is usually used in a concentration of 2 to 3%, and there are two types of agar that is called as New Zealand agar and Japanese agar, which is obtained from the seaweeds. So a New Zealand agar is much more being used than the Japanese agar. So most commonly it is जो most commonly used agar है वो New Zealand agar. Why it is preferred over Japanese agar? Because it has more gelling capacity. It has more gelling capacity. So and the agar is available as fibers, shreds or as powders. So physical factors जो कि related है liquid जो कल्चर मीडिया होता है उसमें कोई भी अगार नहीं होता एंड इट इज कॉल्ड एज ब्रॉथ. फॉर एग्जांपल यू कैन सी इन द डायग्राम दैट देयर इज एन ब्रॉथ ओवर हियर. In the solid media, the concentration of the agar used is 2 to 3% agar. As you can see that I have mentioned an agar plate over here which is solidified. And semi-solid agar, it consists of 0.2 to 0.5%, and it is basically a semi-solid agar, and it is basically a semi-solid agar, right? So, and this agar is used for the motility testing. So basically, now we are discussing the liquid media. Liquid media itself means that there is no agar present. Its uses are that it is used in the processing of routine cultures, biochemical testing, presumptive bacterial growth, for inoculum preparation, and for isolations of pathogen from a bacteria. The most common liquid media used in the microbiology lab are the nutrient broth and nutrient broth. Basically, we can see in the picture that there is a nutrient broth, and if there is an organism inoculated in the media, it shows turbidity. There can be sediment also, and there is a pellicle formation also, which is basically seen in Pseudomonas aeruginosa, Vibrio cholera. These are the organisms in which the pellicle can be seen. So now the next question is, what is the pellicle? Pellicle implies the multifaceted response to the changing environment which is induced by the bacterial growth, and it is formed between the air and the liquid interface of the standing liquid media. That means air and the liquid interface जो होता है, that means at the top of the media, इसके ऊपर आपका pellicle formation होता है. Now we are going to discuss solid media. तो solid media itself means कि इसमें agar है. Advantages इसके ये होते हैं कि we can distinctly see the colony formation, colony morphology. We can see the pigmentation, hemolysis if it is present, quantitative bacterial count, and we can easily isolate or we can detect if there is a pure culture or a mixed growth. For example of solid media are nutrient agar, blood agar. All these are the examples of solid media. Now we are discussing the types of culture media. In this, we will discuss the nutritional factors according to the nutritional factors. For example, simple media, complex media, synthetic media, and the special media. under the special media, there are many, for example, enriched media, enrichment, selective, differential, transport, and sugar fermentation media. Now, depending upon the presence of oxygen. Depending upon the presence of oxygen, the media can be aerobic media. That means the organisms will grow in the presence of an oxygen, and anaerobic media in which the organisms will grow in the absence of an oxygen. So here this is the picture presentation. The media which is present in a tube, for example, a liquid media, the upper part of the media, the pink part, that means it has been exposed to the oxygen, so it is called as the oxic zone. Whereas below the pink part, it is the representation which is showing, which is not in contact with the surface or the oxygen, it is called as anoxic zone. Now, simple basal media. Simple basal media consists of peptone water, 1% peptone, 0.5% NaCl and water. Its uses are कि ये help करता है to grow most of the pathogenic non-fastidious organism, making hanging drop motility, to test the indole production by putting in Covax reagent, to grow bacteria for antibiotic susceptibility testing, and the sterilization method used is autoclaving. Now the nutrient broth, which I have already discussed previously. So it consists of peptone water and meat extract, and its uses are growing most of the pathogenic non-fastidious organism and studying the bacterial growth curve. So its sterilization is done with the autoclaving. Now nutrient agar, it is a simple basal media and it consists of nutrient broth in which the 2 to 3% agar is added. इसके uses है कि it helps to grow most of the pathogenic non-fastidious organism. It is used to study the colony morphology of the bacteria. Pigment demonstration is very remarkable for nutrient agar. Why? For example, in Pseudomonas aeruginosa, the pale color of the nutrient agar will change into light green or according to the pigment. It helps for performing the biochemical tests also. And the method of sterilization used for it is autoclaving. Now the complex media. All medias except the simple media are the complex media. Synthetic media. Pure chemical structure and the exact composition of the media is known. For example, the Dubose media with tween 80. Now we will discuss enriched media. Enriched media is in which there is a basal media, for example, the nutrient agar, and additional nutritional requirements like blood and serum is added to it. That is called as enriched media. Example of enriched media are blood agar, in which nutrient agar is used and 5 to 10% of the sheep blood is added at 45 degrees centigrade. Its uses are it supports the growth of the most of the bacteria and it helps to demonstrate the hemolysis, which can be partial, alpha, complete or beta hemolysis. It can differentiate between the different various gram-positive cocci on the basis of hemolysis. And the sterilization is basically it is of nutrient agar, which is done by autoclaving, into which aseptically defibrinated 5 to 10% of sheep blood is added at 45 degrees centigrade. Now the chocolate agar. So here also the medium will be the same. Basal medium, simple media, for example, nutrient agar, and 5 to 10% sheep blood is added at 70 degrees centigrade. That means the temperature has been raised from 45 in the blood agar to the 70 degrees centigrade in the chocolate agar. So the RBCs जो होते हैं वो lyse हो जाएंगे and उसमें से contents जो होता है वो release हो जाएगा. For example, hemin factor and factor 5. So uses are more nutritious than blood agar and supports the growth of the fastidious organism like Neisseria, pneumococci and hemophiliacs. And the sterilization Hemophilus influenzae, and sterilization is done by autoclaving. Now we will discuss the Loeffler serum slope. Solid medium without agar and it contains the serum which acts as a solidification agent. So it helps to grow the Corynebacterium diphtheriae, and the sterilization, all the contents are sterilized separately and then mixed. Solidification of serum is done by inspissation. So inspissation helps in the solidification of a media. Now students, I will tell you something that enriched, when I'm talking about enriched, it means that it is a solid media, and when I'm talking about enrichment, it means that it is a liquid media. Liquid media added with an inhibitory agent to suppress the normal flora and stimulate the growth of the pathogenic organism. For example, tetrathionate broth, which is used for the salmonella from the stool, gram-negative broth, which is used for the shigella from the stool, selenite F broth, that is used for the isolation of shigella from stool, and alkaline peptone water, which is used for the isolation of Vibrio cholerae from the stool. Here students, I would like to speak again on this thing and stress on this thing, that enriched media, remember this thing, it is a solid media, whereas enrichment media, it itself means that it is an liquid media. So now we are discussing the selective media. Selective media, that means it promotes the growth of a certain bacteria and inhibits the growth of the other bacteria. It is a solid media which is added with an inhibitory agent to suppress the normal flora and stimulate the growth of the pathogenic organism. For example, deoxycholate citrate agar, XLD agar, that is called as xylose lysine deoxycholate agar, which isolate the salmonella and shigella from stool. There is another selective media which is called as potassium tellurite agar. It is used for the isolation of Corynebacterium diphtheriae. Then there is a PCBS agar, that is called as thiosulfate citrate bile salt agar. It is used for the isolation of Vibrio species. Sterilization method is same, that is called as autoclaving. So next is the Lowenthal-Jensen medium. It is a solid media without agar and it contains egg. And the solidification of the egg is done by inspissation. Okay? So malachite green, it is a green selective agent and it is added, it is basically green which is added to inhibit the microorganisms other than the mycobacteria, and it also acts as a pH indicator. इसका यूज क्या होता है लॉवेंसन जेंसन मीडिया का? It grows the Mycobacterium tuberculosis and atypical mycobacteria, and it takes approximately 3 to 6 weeks to grow, and for antibiotic susceptibility also for the Mycobacterium tuberculosis.